So I'll try to keep it interesting and try to keep you a little bit awake during the presentations. All right, let's talk about what is plant breeding. And there are two definitions that I like. I started out, I was a professor here for 17 years and taught this course, in fact, way back before a lot of you were born probably. And sort of the academic definition of plant breeding is the art and science of improving the heredity of plants for the benefit of humankind. I left here in 1987 and spent 12 years managing research and development for Cargill hybrid seeds worldwide, and so worked in industry research management and learned that the definition had to be modified. Plant breeding is the science, art, and business of improving plants for human benefit. Or in fact, as the seed industry would say, the science, art, and business of improving seeds for profits. Overall, about 500 plant species have been domesticated, and that's out of over 1 million species that are possible. By domestication, that means they've been brought under cultivation. And we'll point out, people tend to think that when man arrived on Earth, that there were lots of crops from which to select for food and development. It didn't quite happen that way. We'll talk today primarily about the areas of crop domestication, what I call classical plant breeding, which started with the rediscovery of Mendel's laws in the early 1900s up till about the 1990s. Then I'll talk about plant breeding today from about 1995 onward, where in addition to the applied traditional breeding, there are a lot of tools, molecular tools that are available to make plant breeding more efficient in ways and hopefully more productive. So here, this is from Guns, Germs, and Steels. People invented agricultural technology. I mean, before mankind, there was no agriculture. Natural selection worked on all species on the planet, but natural selection did not select certain plant types to be more or less appealing to humans. People did the selection. That started about 10,000 years ago. Since then, people have invented various farming systems. I mean, people invented conventional agriculture technology, and they invented organic agriculture technology, and they invented biotechnology. So none of these were sort of natural systems. All of these are man-made, man-invented systems. In terms of crop evolution, humans have always guided the evolution of crops. A small sample of wild plants were chosen from thousands of wild species and domesticated. They've undergone 10,000 years of selection. And most importantly here, all crops we grow today were once wild plants. They were selected, domesticated, and developed by humans. More importantly, none of those crops would survive in nature today if humans disappeared from the planet. So the system of agriculture has lots of advantages and lots of disadvantages, but for the most part, our system of agriculture was created by humans in order to provide food, clothing, and shelter for humans and animals. As you might guess, the crops, the various strains, and the genes that are contained therein have moved around the globe with humans. As early human populations migrated around the globe, they carried the plants with them. So domestication has done things like, this is sort of the progenitor of corn, teosinte, and over 10,000 years with domestication and some classical breeding, we've modified that to maize as we know it today. These are the progenitors of tomato. And in fact, these are actually fairly toxic plants, and they were selected, domesticated, and enlarged to produce the tomatoes that we know today. In terms of where all of this domestication took place, the Russian scientist Vavilov has come up with 8 centers of origin. And so for years, we sort of looked at the map and says, okay, and this is a little bit confusing. Some people say 8 centers and some 11. But you notice Vavilov has a 2 and a 2A, and he has an 8, an 8A, and an 8B. So 8 to 11 centers of origin. And the concept was that within certain centers, human populations selected and domesticated different plant crops. And it was a fairly simplistic look at how the crops evolved with the developing human populations. And it led to lots of arguments as, well, wait a minute, what do you mean that sorghums started out over here in China or India? Didn't they start in Africa? Well, in fact, just a list of Vavilov's centers. Yeah, in fact, the origin of crops were not nearly as discrete, and they couldn't really be classified and set in geographies. It's a little bit important because I've seen over the years graduate students say, okay, I have a disease of this crop of interest, and if I really wanted to find genetic diversity for resistance to this disease, I'll go to the center of origin of my crop. And sometimes that's very sound reasoning, and sometimes it works. Other times it doesn't work. You need to go to the center of diversity or origin of your crop, but you also need to go to the region where your crop developed in association with a pathogen. So back in the early 70s, Jack Harlan changed our views on domestication and evolution of crops. Jack says there really weren't any major centers of diversification. He says there were some centers, but then also sort of larger diffuse areas that were non-centers. And so certain crops were developed in the Near East, but really all through from the Near East down through Africa, these crops were sort of modified, and they were secondary centers of domestication. And he lists 3, the Near East with Africa sort of getting the overflow, China with Southeast Asia sort of being the diverse centers, Mesoamerica with South America sort of carrying some domestication traits. Well, since then, evidence says these centers are more diffuse than even Harlan had recognized, and that in fact, most of our crops may have had one primary site where domestication started, but lots of secondary or tertiary sites where it continued. And then in fact, very early on, these crops started moving all around the world. And how would you think the crops would move around the world? With people. As humans moved around the world, they would carry their crops with them, and they would modify and select their crops as they reached different environments and ran into different spectra of disease and insect pressure or different environmental stresses. So the best we can do today is say, well, here's some probable geographic origins of some of our crops. Although most of these crops probably moved to other places and were selected as different localized gene pools in those places. And I won't go through this list. A lot of this material I just put in case you're interested for reference. You can pull it out. It works great on if you get into plant breeding trivia games. You can pull this stuff up and look like a whiz. There is one table I'll put that sort of major crops around the ancient world. And the best I can tell, ancient world is sometimes BC. How far back BC, this table doesn't say, but it sort of says that back at the time when humans certainly were had settled in the Fertile Crescent, China, Mesoamerica, Andes, West Africa, India, Ethiopia, United States, and New Guinea. These are sort of the foods that they had. And this is sort of based on a combination of archaeological and sociological data and inferences. And it's kind of interesting, and I used to think, well, this is kind of neat because there's no rice in India. But, you know, recently, Luis's major professor, Dr. Susan McCooch, is convincing me that, well, probably rice didn't originate exactly in India. Probably China and Southeast Asia with secondary origin in India, or maybe even more likely Bangladesh. I just throw these next two slides up just to sort of give you a perspective that over this time that all of these crops are being domesticated. I mean, you know, in general we think, well, this is thousands of years. So if you were going to start today to develop a new crop, it's going to take you, what, 8,000, 10,000 years to get it done? It's not something that you'd want to choose as a thesis project. But actually, when we look at this, the timeline's a little bit deceptive. And I put this up primarily to show, going from 12,000 years ago to 7,000 years ago, the population of the Earth was about 5 million. I mean, so populations could move around, they could produce and grow their food, and the concept of population pressure and food security just didn't exist. I mean, there were more imminent dangers to human population survival than food security. If we take this forward, starting 6,000 years ago, the human population jumped up to 7 million. Look what it did as of 1,000 years ago. 245 to 345 million people. And so during this time, human population started on that exponential increase, and the numbers of new crops certainly didn't go up exponentially, but the production of each of those crops probably went up enough that the world could very easily feed 350 million people. The point I'm making is, in fact, when you look at the genetics of crop domestication, it's a pretty simple study. I mean, you could, in fact, do a PhD thesis on the genetics of domestication of your favorite crop. Because for the most part, there are very few genes, or in fact very few regions in the genome involved in domestication genes. Several of these genes have major phenotypic effects. The genes for domestication represent a very small subset of the total genes within a crop's genome. So theoretically, domestication could occur quite rapidly. And the reason for it taking apparently thousands of years was the fact that the domestication was linked with the development and evolution of human societies. As humans went from nomadic tribes wandering and primarily hunting and fishing and then producing crops to more settled communities planting and growing crops, the domestication sort of followed along. So in point of fact, if you went out and find some wild plant growing on the roadside today and decide, well, I want to do a PhD thesis on domestication of this, and I'm going to make my own crop. Theoretically, at least, you could do that, and you could do that in relatively short time. Sort of a side effect of this, though, is during this domestication process, even though it only involved a relatively few genes within the crop's genome, there was still loss of genetic diversity. I don't know how good your feeling about genetics is today, whether you've had genetics recently or not or never. But if you remember back to your genetics, you remember the phenomenon of linkage. And so even though there were only a few genes for domestication, often when you selected for a gene that equaled a domestic crop versus a wild species, you brought other genes along with it, and you excluded genes that were not linked. So as we domesticated, we sort of pushed our crops through the first little bottleneck, and we lost some diversity. So wild relatives of crops are still important reservoirs of genetic diversity. Okay, traits associated with domestication. Seed size. I mean, for a plant species to survive as a wild species, all it needs to produce are a few seed, and they can be very, very tiny. For humans to use that plant for food, we would rather have bigger seed. So the selection for seed size is one of the earliest traits associated with it. Seed abundance. Here we go back to Teosinian corn. I mean, Teosini survived as a wild species. It still survives today as a wild species. It produces a few seed, and it distributes those seed, and the next generation those seed grow, and so the species can maintain itself year after year after year. Well, humans would go hungry harvesting the yield from a field of teosinte today, but selecting maize with a lot more seed per ear is a trait that humans favored and developed, selected for. No shattering. For the wild plant species, you know, shattering refers to when the fruits mature, when the pods, in this case beans, mature, they open and the seeds shatter and spread out over the ground around the plant. Well, for the plant species survival, that's very good. It ensures that those seeds don't stay locked in that fruit case and rot. They shatter, they hit the ground, they can germinate and regrow. Well, again, for human populations, for rice or sorghum or any of our pulse crops or beans, it's desirable to keep those seeds in the pod. I mean, we like to go out and harvest the fruit and have the seeds still born within the fruit. Thinner seed coats. I don't have a picture of that, but basically, thinner seed coats made the seed more digestible and more palatable to humans. Also makes it more digestible and palatable to other animals. And in fact, wild plants in nature would often have their seeds not as digestible. You know, they don't mind animals eating the seed and carry them across country, but they would like for the seed to come out of the other end of the animal in a relatively intact form so it could germinate and preserve the species. Uniform germination and ripening. For the plant's point of view, a wild species, it's better to have seed germinate, new seedlings germinate over a wide time period, because you're more likely then to hit a favorable environmental regime when you germinate. And the same thing, it's better to have your seed and your fruit ripen at different times, just so that you can buffer against environmental variations. And of course, flavor. I mean, natural selection on wild plant species, there's absolutely no selection for flavor. If anything, the selection for flavor with animals, predators eating the seed, is to select against, you know, not very good tasting and often toxic materials to prevent that seed from being chewed and eaten. All right, so through this process of plant domestication, over 10,000 years, we've gone from wild species to domesticated crops. You'd like to think that that process probably could continue. I mean, who did these selections to domesticate the crops? What kind of people did these? What do we call these people? Who grows these crops today? Farmers. So basically, starting 10,000 years ago, farmers started to select. They would pick characters they liked, and they would save the seed, and they would take that seed with them and plant them in the next cycle. And so a lot of people argue today, well, I mean, these guys have been going well for 10,000 years now. I mean, look at the gains they made in the productivity of all those crops. Well, yeah, they made some gains. And today, yeah, farmers still select and play a very important role in selecting local varieties and local land races. The one thing that's missing in most of the farmers' selection is they can take whatever genetic variation appears before them and select the best types, but they can't create any new genetic variation. So starting about 100 years ago, when we learned about Mendel's laws and genetics and how all these traits are inherited, and the fact that we can sort of control our own destiny, our own evolution, classical plant breeders started first finding or looking for genetic variation among our crops, but more importantly, creating genetic variation. Then they would evaluate the variation over different environments. They would select the improved types for specific farming systems and desirable traits. And that had to go hand in hand. I mean, as early plant breeders started working, they selected varieties that would perform under current farming systems. As agronomy and soil scientists worked with farmers to improve farming systems, well then plant breeders in turn selected new varieties that would fit newer farming systems. And so this process has gone hand in hand for the last 100 years. Finally, the classical plant breeding develops and provides farmers with improved varieties. And we'll talk about all of these various steps and concepts in a lot more detail. In fact, starting with about a week's worth of review of the genetic principles that underlie this creation and selection of variation. So what has classical plant breeding done over the last 100 years? It's increased crop productivity, dramatically increased productivity at a rate that is exponentially higher than what happened the previous 10,000 years through farmer selection. They developed and introduced hybrids. And we'll talk about, in fact, we'll have an entire lecture on hybrids and the concept of heterosis and hybrid vigor. Developed and introduced resistance to diseases and insect pests. And today I'm almost to the point of saying introduced resistance or tolerance to environmental stresses. We're getting to the point where there are actually some very useful materials for environmental stress resistance or tolerance. They increase food quality and quantity. And they develop crops adaptable to mechanical harvest. Why was that necessary? This is a quiet group.
Go from harvesting with the hands to large combines going through the fields, and you need things that are level so the combine obviously gets all the help it wants.
All right, so in order for a crop to be adapted to mechanical harvesting, it's got to be pretty much a level field, fairly uniform. It's got to mature at the same time. Okay, but if we develop a uniform field of corn that's grown in Iowa and can be mechanically harvested, doesn't that eliminate a lot of our genetic diversity? And doesn't that make us a lot more vulnerable to changes in environment or diseases or insects? So why did we go to mechanical harvesting? Why mechanical harvesting? For what? For convenience. So, well, just for convenience?
Well, I mean, it makes it easier, more money, all the associated benefits of having the...
It's easier, it's convenient, it's more money. I mean, I worked a lot today in Africa where most of the harvest is still by hand. I mean, in Africa, there's still labor available, although the cost for using that labor to harvest those crops is very, very great in terms of the children not being able to go to school or the populations being tired and diseased. There's a lot of cost. So it's convenient, it's easy, but the basic point is we had to have more food. We needed more food, and we needed more food produced per acre or hectare of land, and we needed that food produced cheaply and efficiently. And so mechanical harvest is really one of the things that drove the agricultural revolution. Unfortunately, along with the plowing and the additions of fertilizer and the mechanical harvesting, we created a system of agriculture that's not very environmentally friendly. I mean, no way you can say that farming is environmentally friendly. So why on earth do we let people farm? That answer will become more apparent in about 3 hours when your stomachs start rumbling and you're ready for lunch. Because we get hungry. We like food. And if we're going to produce enough food for all of the people on earth today, unfortunately, we're going to do it through a fairly extensive and environmentally unfriendly, in some cases, agriculture. Has classical plant breeding worked? Here's the yields, relative yields from 1930 to 1990 of a whole series of crops. You'll see I'm particularly proud. Maize and sorghum, really, the yields have shot up. Demonstrating the concept, and sorghum shot up in the U.S. pretty much along with the switch from pure line sorghum varieties to hybrids. So it shows this concept of heterosis hybrid vigor really works. Interesting enough, today rice is beginning to take an upturn, particularly in China, as rice production is moving more to hybrid production, at least in China. Not much anywhere else. But basically, whether we're dealing with hybrids and hybrid vigor, or whether we're dealing with increase of varieties, such as peanuts and wheat and rice, we've seen a steady increase in food productivity. Now, basically, the rule of thumb says 50% of that is due to better genes in the crops, and 50% due to improved cultural practices. But as I said before, as the cultural practices changed and improved, the plant breeders must select for varieties that fit those practices. Also had a green revolution for wheat and rice. I don't know, a few years ago, everybody coming through this class knew the green revolution. Today, it's kind of ancient history, back in the mid-'80s. But basically, this shows the increase. The darker color is rice, and the lighter color is wheat. The white bar is the yield in 1965, and the colored bar, the yield in 1985. And it shows, number one, the increase in productivity over that 20-year period. For the countries in dark green were the primary countries that benefited from the Green Revolution. So the Green Revolution in itself, it lifted billions of people out of poverty. Well, it did. It changed the undernourished rate from 38% to 19% in a 20-year period. Food consumption per capita was increased everywhere except in Africa. Food production in India went from 50 to 205 million tons over 5 decades. Wheat from 60 to 82 million tons per year. So there was less starvation and famine, increased food sufficiency. 4, these countries, at a time when starvation was the major threat for India, Pakistan, China to a large part, for survival for individual people, the major threat for world peace and world security. in terms of people getting hungry and deciding that India was going to conquer Pakistan, or Pakistan was going to conquer India. And the Western Bloc was going to help India, and the Soviet Bloc was going to help Pakistan. And so this was at a time where people were starving and things were desperate. Unfortunately, you know, there are side effects of the Green Revolution that really nobody could foresee. The fact, and let me go to the next slide. The fact that the Green Revolution focused on a few crops, wheat, rice, and really there wasn't much of a Green Revolution for maize. I mean, it's got some increase in maize production, but primarily wheat and rice, meant that in large parts of Asia, agriculture went from a very diverse agriculture, growing vegetables, pulse crops, along with the cereals, to pretty much a monoculture, growing wheat or rice. And so today we face problems with nutrient deficiencies, particularly micronutrient deficiencies through those areas where people are eating primarily a rice diet. It's elected for response to improved management, including fertilizer and pesticides. And at the time, fuel was cheap, nitrogen was cheap. It was a logical thing to do. You need more food, pour on the nitrogen, increase the food. Well, over the 20 years that the Green Revolution was working, the prices of fuel and therefore nitrogen began to increase. So in a lot of these countries, it became a dilemma. How can I grow these new improved varieties because the individual farmers can't afford the credit to get the fertilizer and the pesticides? And so there were a lot of secondary problems. But, you know, the way I feel you have to look at it is there are hundreds of millions of people in Asia alive today who are fortunate to face problems of lack of income for nitrogen and nutrient deficiencies, because had it not been for the increased food production, those people wouldn't be here today. The populations of those countries would have either been lower, or else those countries would have burst out over their borders and their populations would be living here in Ithaca or somewhere else in the world. So crop yield was the major goal. Very successful in Asia, was not successful in Africa. And we can talk, in fact, later we'll do a whole class, I think, if I get to that, about why it's not successful in Africa. I mean, basically in Africa today, the environments are so diverse, the cropping patterns are so complex, and yield is not the major driving factor. Quality and acceptance of the food that you produce within the diets and within the local societies are much more important. And even though Africa has problems with population pressure, and we certainly have lots of starvation, a lot of our problem in Africa is not just revving up the amount of yield. It's getting production into a better form of varieties that the farmers and the local people will grow. All right, so starting about the mid-90s, new tools were developed. Well, tissue culture tools have been around for 50, 70 years. Recombinant DNA tools really were the ones that started up through the mid-90s, and transgenic plants particularly in the mid-90s. Just a preview, we will subsequently have lectures on tissue culture applications and transgenic plants and so on and so forth. But tissue culture really provides plant breeders with two major things. First, it helps to select and manage superior plants so that if you can produce a good variety and it's a cross out-pollinated plant and you can't really increase that through any sexual cycle to maintain that pure variety, you can go through vegetative propagation and utilize tissue culture techniques to maintain and manage individual genetic packages. More importantly, tissue culture gave breeders a means to expand the gene pool. I pointed out that breeders not only find genetic diversity, they create diversity. So somoclone variation, crosses between wild-related plants aided by embryo culture, somatic hybridizations to actually fuse cells, and then regeneration of fertile plants after transformation all broadens the gene pool and creates diversity. Recombinant DNA tools allow us to identify genes that control traits and to move those genes around to produce crops with improved traits. Probably the biggest application to date in plant breeding is marker-assisted breeding, and we'll talk about that extensively in another lecture. The use of molecular markers. They're located in known positions in the genome. They're easily assayed with small amounts of tissue. Occasionally, they happen to be located next to a gene of interest. And then plant transformation technologies basically allow us to insert one or a few genes into a plant. We can take genes from distantly related species, from bacteria, for instance. It's a relatively precise process, not as precise as my colleagues down the hall would like to have us believe, but relatively precise. Point is, once you develop transgenic plants, they still need to be evaluated and tested. To show you just a little cartoon on that, and we'll talk about this probably the next lecture, about genes being located together on chromosomes and linkage. So using traditional procedures, you take a blue-beaded chromosome and a green-beaded and a blue-beaded, and you get a mixture. And you sort out for years and years and years to get back to one type of trait inserted. If you use transformation technologies, you can just take one gene, put it into the location somewhere in your recipient chromosome. That's sort of it for today, and since we're out of time, I will just bring your, oh, excuse me, losing my voice. Bring your attention to there are other slides on this deal. This sort of shows the incidence of hunger, malnourished children, but the major point of this whole next part is even today, with all of our technology and all of our improvements and all of our selections, what does mankind eat? Well, we eat rice, wheat, corn or maize, potatoes, millet, sorghum, cassava, rye, barley, and then other things. And so our major diet is consumed of rice, wheat, and maize. And when you add in the fact that maize is also fed to animals, those three crops are sort of the mainstays of our world food supply. And just to show one other slide, you can see population is increasing. It's actually not increasing as fast as it has been. Cereal production is increasing. So you say, well, in the future we don't have a lot to worry about because we're producing more cereals. That production is going up faster than population. But a large part of that cereal production is maize, and it's going into meat production. Which would be nice to say, that's great, that's more food. But when you look at that on a country-by-country basis, it means that certain countries like India and Thailand and China and South Africa and Brazil and Argentina are moving through the stage where people are not just eating rice or maize or wheat or potatoes or cassava. They're eating meat, so they're using more and more grain for more and more meat production. And China particularly, the need for grain for meat production in China over the next 10 years is going to go up exponentially. There are probably today more chickens in China than people. and there will soon be as many hogs as people. And so the meat productivity is increasing. Some references on that part, and then just the last slide just gives you some references of who breeds crops today. Universities, the CIGAR centers, we'll talk about those in another class. National research programs in most developing countries, and the seed industry. I didn't put a link because the national research programs, you could maybe pull up links for Africa, Asia, Latin America, but they're so numerous and so many there that the best bet is just go to any country you're interested in and plug in NAR, NARS, NARE. It's National Research Extension, but NARs will get you to a series of linkages. Are there questions? All right. Well, on Monday, I will give you a chance to think over what we've covered today, and if you have questions before we get started in the lecture Monday, I'll take those. Otherwise, please leave the little information half page with us, and we'll see you